want to give you a bit of background to the case study I'm going to talk about. Basically, back in 2008, we published a paper on the vulnerability of marine invertebrates, excluding corals, on the Great Barrier Reef to climate change. And we used a variety of scenarios that were given to us by CSIRO. This was originally a contract for Gurumpa, which produced a very large volume on the vulnerability of the GBR to climate change. We raised this whole paper raised various scenarios, such as, does actually the data exist to support them? So after we'd published this paper, a group of us said, right, okay, let's see whether we've actually got the data to support some of the predictions that we made, that species are going to go extinct or species are going to move south, etc. So we applied two approaches. We undertook a global meta-analysis of museum records, focusing on marine studies. We found that 13 satisfied our stringent criteria, representing 311 species. We looked at regional studies, and we looked initially at the mollusk data from both the Museum of Victoria and the Australian Museum. Why did we choose mollusks and those two institutions? Was that there's been a long history of mollusk collecting at those two institutions. Plus, there are the relevant specialists for us to go and talk to. The regional studies, and we had a very large number of records, and something like over 5,000 records from the Queensland-New South Wales border down to Bass Strait. But of those large number of records, only relatively few had sufficient numbers of individual species. So only 207 of those species had 4%, had enough records to even look at, and only 53 had only more than 200 records. So the data was fairly scattered. We had a lot of species and a lot of records, but we didn't have consistent records for those species over time. So how do you get around this? And we were working with Mick Ashcroft, a GIS expert here at the Australian Museum, and so we decided to select species occurring around Sydney to look at whether we could detect species that over time had moved south or moved north. And we chose species that had over 10 records before and after 1970. Why did we choose 1970? We really chose 1970 to incorporate the increased rates of warming which have occurred over the last 60 years. And it suggested that the increases in temperature is something like 1.5 degrees. The results of the meta-analysis, and as I say, this has been published, showed that marine data of those 13 studies have actually shifted southwards. And it was present in both all groups and within individual groups and in all regions of the world. And those were the sorts of rates that we found based on that meta-analysis. So quite a substantial movement. So we're suggesting that based on that, marine invertebrates are actually shifting polewards on a global scale. And the species in those meta-studies, in those 13 studies, had a range of distributions, i.e. intertidal, shallow, subtidal and deeper water. And they also had a variety of dispersal potentials. Some were spawners, some were brooders. Now, I'd now like to go on to the results of the regional studies where we use the records from the Australian Museum and the Museum of Victoria. So basically the results showed a slightly mixed response. Some species had moved northwards and some had moved southwards. So it was a very confusing pattern. So why are we getting such conflicting results using museum data? Taxonomic inconsistencies, there's still some taxonomic revisions required of some of those species we selected. There's also been a considerable amount of reduction in collecting since especially the 1980s. Part of this is a result of reduction in field trips by the experts and increasing regulations in actually the ability to go out and collect. This is meant with marine parks, this is meant basically we've lost the amateur shell collectors. And the collecting that has been done more recently has been highly targeted based on the research interest of the relevant researchers. So one of my take-home messages is that in order to interpret the results you get from both the wide-scale and the regional studies, we need to have experts to the relevant experts. Not only the researchers, and I think it's fair to say that Winston Ponder and Ian Locke and Bill Rudman here at the museum, together with the collection managers, especially Ian and Des Beachy, were incredibly useful in helping us to analyse that data. What is the way forward? I think that we need to really, in future, look at species, preferably perhaps large species, easy to identify with fully resolved taxonomy. We need to look at the specimens on the boundaries of their distributions to see whether they are actually mature and breeding rather than just vagrants. I know that a lot of people said, oh, those species are vagrants, that's just an odd specimen, it doesn't mean anything. So really we need to check whether or not they are breeding. And we also, in order to work out which species we need to look at, we need to involve the relevant experts. And I would suggest, and we did in this paper, that we should set up permanent transects along the New South Wales coast intertidally. And perhaps we should go out and regularly sample for those selected species every 5 or 10 years for the next, hopefully 50, 80 years. And I think we will see dramatic changes occurring. But combined, and I think Craig alluded to this, it's not just the distribution of those species. We also need to look at their physiology and seeing how they're adapting. Are those species that are successful in moving south or north coping with the changes in climate? What are the complications, especially of the marine data? I think that the range shifts are not as obvious in marine as in terrestrial systems. This is probably, well, I'm sure it's due to the open nature of marine systems and the multiple scales at which environmental factors operate may mask climate change. Recent studies have shown that intertidal fauna, which is what we concentrated on, are driven more by oceanography and geomorphology than by temperature. And we, as I say, we know that climate change is not just increasing temperature, but it's changing the whole the frequency and the intensity of storms, and changes in currents are occurring. And this is occurring on a very local scale, and it's not easy to predict. So in conclusion, museum collections do have a role to play in understanding how climate change is affecting our marine ecosystems, but that data needs to be carefully scrutinised and interpreted by the experts. And that's, when I say experts, that's not just the researchers, it also must include the collection managers, because they have a wealth of information as to how that data was collected and who collected it and why they collected it. And then we need to look at that data and analyse it with regards to the biology of those species. And that will also work out which species we need to be targeting and then going back to our collections. Thank you.